A cheap decades-old malaria drug made aged female rats live longer. But those same rats also ate about 10% less food. And eating less, on its own, already lengthens a rodent's life. So this study can't yet tell the drug apart from the smaller meals. The researchers gave aged female rats a drug called chloroquine, a cheap malaria medicine that's been around for decades. They gave it on an on-again, off-again schedule for most of the rats' lives. The treated rats lived longer. The very longest-lived ones reached the end of life about 19% later than the untreated rats. A real lifespan result, on a pill you can already buy, is a rare thing. A longevity drug, we assume, switches the cell's cleanup on. That cleanup is called autophagy, the way a cell breaks down and recycles its own worn-out parts. The best-known longevity drugs turn autophagy up. Chloroquine does the opposite and jams it. So a drug that jams a cell's recycling and still extends life runs backwards. And the authors say they don't yet know how it works. Here's the fact that changes everything. Across decades of animal studies, the single most reliable way to make a rat live longer is to feed it less. Researchers call it caloric restriction, and it works so consistently it's the benchmark every longevity treatment is measured against. Now look at what the treated rats did. For most of the study, the treated rats ate about 10 to 12% less food than the untreated rats. The paper records that gap directly. Same chow, just less of it. That works out to roughly 10% fewer calories every day for months. So put the two facts together. The treated rats did two things at once. They took the drug, and they ate about 10% less. So there are two possible reasons those rats lived longer: the chloroquine or the smaller meals. And this study can't separate them, because both happened to the same rats. No group took the drug while eating normally. No group ate the smaller amount without the drug. The authors say it themselves. The reduced eating may have partially contributed to the longevity effects. How much is the drug, and how much is the diet, is still unmeasured. Now the two numbers side by side. The treated rat's maximum lifespan, meaning the age the very longest-lived ones reached, was about 19% higher than the untreated rats. And right beside it, those same rats ate about 10% fewer calories a day. One number is the exciting result, the other is its shadow. This is a lab result in rats, on a drug that isn't gentle. Chloroquine damaged the rats' livers at the higher dose tested here, and in people it can harm the eyes. It is not a medicine anyone should take. So here's the experiment that would settle it. One extra group of rats. Feed that group the same smaller ration the drug group chose, but give it no drug. If those rats live just as long, the credit goes to eating less. If the drug group still pulls ahead, that gap is the drug's doing, and that would be the real news. The team could run exactly this next. It would still need testing in males and other strains, since this group's earlier chloroquine work was in male mice. A cheap old malaria drug made aged rats live longer, but the same rats also ate less. So the answer to was it the drug is not yet. And now you have the one question for every headline that says something helps you live longer. Did the treated animals also eat less?